Hello everyone. Today we will start with the neo-Mendelism next parts. So previous lecture we spoke about the neo-Mendelism, and in that neo-Mendelism we spoke about the incomplete dominance, in which we considered the plant, like the flowers of the plant Mirabilis jalapa, which is also called as a 4 o'clock plant. After that, we learned about their ratio, genotypic and phenotypic ratio, which were coming same as 1:2:1. Today we will continue with the next topic of codominance. Okay, so today we will start with codominance. Now, what is codominance? Codominance means if suppose there are two alleles, okay, suppose there is two alleles, let us consider that there is a capital A and capital A. This is an example, okay. If suppose there are two alleles, these two alleles are expressing together, okay, means like this capital A is also expressing and this capital A is also expressing, means their expression is equal, so they have equal expression. Okay, so codominance is like coordination, like one person coordinates with the other person, right? Means they are expressing together, they are working together. It's not in coordination what we say that everything will be working, will work together, right? In the same way, in codominance, both the alleles will show equal expression. What will happen? Both alleles will show equal expression. So these two alleles will show what? Equal expression. During which position, like they will show equal expression in the F1 hybrids. Okay, they will show equal expression, they will coordinate with each other and equally express in F1 generation, or you can say during F1 hybrids. Okay, so here there is a case where the alleles are able to express themselves, means capital A is this capital A is showing its own effect, this capital A is showing its own effect. So here we can say that both the alleles are independently expressing. Okay, so they are independently expressing, so these two alleles are independently expressing. Okay, they are independently expressing. So this you will understand when we will take a proper example. So such allele which will express independently and their expression will be equal expression, such alleles are known as co-dominant alleles. What we say? We say it as co-dominant alleles. Okay, we say it as co-dominant alleles. This is about codominance. Starting, just I have started expressing about it. Just started teaching, I mean, expressing means to teach this topic. So in codominance, both alleles are expressing, in short. Likewise, what we studied in the previous class, we studied about the incomplete dominance. So in incomplete dominance, what was happening? We took the example of capital R, small r, okay, which actually came from capital R, capital with this is a red flowers, right? And small r, where small r is showing white flowers. Okay, and then we got F1 as capital R, small r, and this F1 was neither red nor white, but it showed some intermediate color which is pink. Means what? Here the alleles are like not expressing themselves. Means both the alleles are like, means they are in pair. Actually capital R should express its own color red, but it was not expressing, and hence like there is something incomplete. Now you just tell me, when you take red color and pink color, when you add this in a petri, when we do painting, then we get actually pink color, right? We get pink color. if you add if you add red, okay, you add red and you take pink color, okay, so after adding these two colors, red and pink, what we get pink color, right? We will get which color? We will get pink color. In the same way here also we got pink color when red flowers, okay, and white flowers were crossed. Okay, now I will write white properly, like I will highlight it with white, some gray color. So this is white. So white and red, they actually, what they gave us a pink color, right? It gave us a pink color, but both the alleles didn't express completely. So this was about incomplete dominance. What is this? This is incomplete dominance we studied. Now in case of codominance, what is happening? In case of codominance, the situation is that these two alleles both are expressing together. Okay, means both are ready to express together. That also together means both will show its own effect. Both alleles will show its own effect. Let us come to the actual example. Now the example which is there for codominance, there are two examples. Okay, the first example which I will take is like in your textbook only one example is there, so let us take that example only. So the codominance example here goes with the cattle coat color. Okay, so example taken is cattle coat color. Now what they are saying, that there is a cattle, okay, which is having, like we take one female and one male, okay, and this female is having capital R as the red color, that is phenotype. I will mention here as genotype word, the same way we have to follow, the way I had taught during monohybrid cross, and here we will write the phenotype. Okay, so this is capital W, which is white coat color. Okay, so this is for red, and this is for white. I hope this much is clear. The coat color which we are speaking of. Okay, then what we have to do? We have to make the gametes. Okay, so let us make the gametes. So here, since there are two R, I will consider only one R. So I will just write capital R. Since here there are two W, I will consider only one W. So that is capital W. Okay, and this what I have written, these are the gametes. Fine. Then there will be what? There will be fusion of gametes. When there is a fusion of gametes, then we get what? We will get capital R and capital W. This is our F1. Okay. And this F1, now what is this F1 and what actually is happening here? So we have capital R and capital W. So here what is happening? The capital R is giving its own expression and the W is giving its own expression. Both are giving their own expression. R is saying, okay, I will give the expression of red, and W is saying, okay, I also want to give my expression. So they both are expressing together. Both are expressing together. This is also expressing, this is also expressing. So there is two effects. One effect is of capital R and another effect is of capital W. Okay, so there are two effects which we can see. So here there is effect of capital R also, and here there is effect of small like we can say capital W also. So what will happen here, if you will see the organism, so suppose this is a coat color, a patch of it, so it will show either patches of, like suppose the patches like this, like suppose it was entirely white, okay, and on top you will have some patches of red, okay, like this here, or something here, here something, here, here, like this. You will get this as one condition. Or the second condition can also be that it is entirely red. What will be the second condition? That it is entirely red, but it is having patches of white. What it is having? It is having patches of white like this. Okay, like this, there is, it is having patches of white and red. So these both condition are capital R, capital W, capital R, capital W. Okay, and this coat color of, like either having background as white, background as white and patches as red, or background as red and patches as white, this is known as roan. These both are known as roan. And what happened? These two roans were crossed. Then what you will get the possibility as, here you will have two gametes, capital R, capital W. Basically, what we have done, we are doing selfing. Okay, what we are doing? We are doing selfing. Here also we can write capital R, capital W, and when we cross it, we get capital R, we get capital R's, capital W. Here we get capital R, capital W, and here we get capital W, capital W. So I will write it now. So first one will be completely red, okay, and this this one will be white, okay, and this capital R, capital W will be half this, half red and half white, okay. So these will be what? This will be having red coat color, yes. This will be these two will having roan coat color, yes, and this will be having a white coat color. So here if you will see the genotypic ratio and the phenotypic ratio, so if you can see that we have one red is to two roan is to one white. So this is which ratio? This is phenotypic ratio. Okay, and if you go with genotypic ratio, so genotypic ratio is, here you get 1 R is to 2 capital W is to 1 capital W capital W. So here also very important point to be mentioned. I will put stars here because this is very important point. Both, okay, both genotypic and phenotypic ratios are identical. Both genotypic and phenotypic ratios are identical. Okay, so this is very important point. I will mark all the MCQs. Now what will come? So the MCQ number 1, which can be asked, that what is, like what is actually the meaning of the codominance and codominant alleles. Okay, you should know that it is giving an equal expression. This can also be an MCQ. You should know that hybrids are showing both the expression of both the alleles. Okay, you should know that here F1, okay, I will mark it with orange. Here F1 is Rhone. You should know that this is Rhone. Okay, here you should know that what are the possibilities of this, red, how many this, all means the ratio. The ratios are important. They can be asked as MCQ. You can also get an MCQ as which of the following have both genotypic and the phenotypic ratio similar. So you can write this as the statement, which is very, very important. I must say this. So I will highlight it. This is a very important statement. You should know it. Even in incomplete dominance, we saw that there was same genotypic and the phenotypic ratio. So you can actually get the question as this, that which of the following have the same genotypic and the phenotypic ratio. Okay, then you will write for both. Both means this genotypic ratio and phenotypic ratio is similar for incomplete dominance and also for co-dominance. Okay, and we had also studied that both these examples are intragenic interactions. You should know this also, that both codominance and incomplete dominance, these are the examples of intragenic interactions. Now intragenic means the interaction is between the same gene. Okay, if suppose this is the chromosome and if this is the gene, okay, which we are talking about, this is the gene. So the interaction is between this only, means among the same gene, or we can say that the interactions are between the same. Okay, if suppose I will draw it here, and suppose you have this as the gene. So the gene has two alleles. So one gene, I'm zooming for you people, having two alleles. Okay, what is this? This is gene, and the green ones are alleles. Okay, so interaction is between two alleles of same gene. This is a very important point. Two alleles of same gene. You should know this. That interaction is between two alleles of same gene. See, I have shown a gene and I have shown two alleles. So the interaction is among these two only. So it is known as, what we will say it as, it is known as intragenic interaction. Okay, this is very, very important thing which I have explained to you currently. So both codominance and incomplete dominance, this can be asked as an MCQ, is intragenic interaction. And what is the meaning of intragenic interaction? I have explained. Okay, moving forward to the next topic, which is multiple allelism and all. Those topics actually I had started and we had done it in a previous lecture. Multiple allelism and pleiotropy was already taught in the previous lectures, so I don't think that there is a need of repetition, but okay, I will take a small glimpse of it. First we will understand about multiple allelism. Multiple alleles. Now what is multiple alleles? When there is one gene, okay, when there is one gene but it has many alleles. Okay, what we say generally, that in general condition, what we say that generally we say that there is one gene and two alleles, but it is found that one gene can also have a number of alleles. A number of alleles. So we say this condition as multiple allelism. Okay, and we know that all the alleles occupy same loci. So these many alleles occupy same loci. What it does, it occupies same loci, same locus we can say. Okay, same locus on the chromosome. Locus means what? See, if this is a chromosome, and where is the location of the gene? Suppose this is a pink color gene, it's present here. So this location of the pink colored gene is on the chromosome. Okay, the location of the location of gene on chromosome is known as what? Locus. Okay, this is the important word. You should know what is the meaning of locus. Yes. So this was about multiple allelism. You should know this much. This is the very important thing you should know. Okay, like I had taken example of Drosophila. Like Drosophila has multiple, multiple. You can say the uh, like alleles for wings. Okay, like if you will go and check there is a VG plus. This is for normal wings. Okay, this is genotype which I have written here. This is genotype which I am writing on my left, and this is phenotype. Okay, so here you have VG plus, then you also have VGNI, where NI stands for nicked wings. Okay, nicked wings. Then you also have VGNO, okay, VGNO, which is notched wings. Then you also have VGST. See, whatever we have written raised to and all, those words are actually the phenotype. This is strap wings. Okay, these are the types of wings. You have VG, only VG, then we just say it as vestigial wing. So these are, and we have beyond this, but for now this much. So these are the genotypes, and this is for what? This is example given for Drosophila. Okay, see if you can see there are so many, but it is one gene with these many. Okay, these are what? These all are alleles. One gene with many alleles. Like in this case, there are many alleles for wing type. Wing is wing is wing is having only one gene, but it has many alleles in Drosophila. Now another example, if I take, then we can take as ABO blood grouping. See if in your textbook they have added comma comma like this, A comma B comma O. No, this is not a right way to represent. We just have to represent it as ABO, the way I have shown here ABO. That's it. Okay, now in ABO, what actually happens? ABO blood group. So we have A blood group. Okay, A blood group. We also have B blood group. Okay, then we have O blood group. These are the four phenotype of blood group. What are these? These are the phenotype. What are these? These are the phenotype. But what if I want to know the genotype? So let's know the genotype of these blood groups. So there is only one gene for blood grouping, one gene, but there will be multiple alleles. Okay, there will be what? Multiple alleles for it. Likewise, in genotype for this, you can write I raised to A, I raised to A. I is a gene. Okay, so these there will be capital A, capital A. Then this will be blood group A. It can also be like capital I. We will write A and 0. If this is a case, then also it will be blood group A. So there are two genotypes for blood group A. And likewise, same you have for blood group B. Okay. And for blood group O, you have this as the genotype. And for blood group AB, you have this as genotype. Okay. So these are the possibilities for the blood group. Not mentioned in the textbooks, but I am just giving you the example and you will solve some questions also based on it. Understood this much? So this is about the genotype and the phenotype, and this is actually example of multiple alleles. Okay, this is example of multiple alleles. So I have taken it. Okay, is it clear? Next, let's move towards some questions. Now, suppose there are two parents having blood group as AO and there is one parent having AB as the blood group. Okay, I will give you 5 examples. If suppose parent A is having BO as a blood group and AB, another parent is having AB. Suppose one parent is having O as the blood group and the second parent is having A as the blood group. Okay, Next we will take as, suppose one parent is having O and the another parent is having BO. Suppose one parent is having O and another parent is having AB. So we will take these all examples. So there will be 1, 2, 3, 4, 5 examples total. So we will write it as IA and IO, because in genotype we have to express like this. Okay, and IA and IB. This I have given you as the question. Okay, this is the first question which I have given. You will write it like this and you will cross. So here you will have two gametes, here also you will have two gametes. So we will directly, we can do here, you will get these two as the one possibility, that is I A I A, and the second possibility will go as I A I B. Next you will have I O I A. Next you will have I O I B. So here you will have the blood group. You have four possibilities. So you will have here the blood group as A blood group. Here you will have blood groups as AB, whatever you have written in race to. Here you will have blood group as A, and here you have blood group as B. So these are the possibilities of whom? Of the offsprings. Okay, possibility for offspring. These are the two parents. This is parents. Okay, parents blood groups. Okay. Next we will go with the same flow as IB. Can write IO and IB. Then you will have IA, IB, and again you will have the possibilities. Okay. So here you will have what? You will have IO, IA. Then you will have IO, IB. Then you will have IA, IB. Then you will have, okay, I have made little smaller arrows, you just adjust this. Then you will have IB, IB. Okay, so we will have here as A blood group, here as B blood group, here as AB blood group, here as B blood group. So we will have four possibilities. So these are the parents, and here you have the four possibilities of offspring. Okay, next, this was a second example. I will move towards the third example. Okay, so this is, we can take only one O, which means what? I O cross with IO IA. Doesn't matter if you don't take these two. Ultimately you will get the, okay, let's take the same if you want. So I naught IA, then I naught IA. So here you will get O, here you will get A blood group, here you will get O blood group, here you will get A blood group. So it's 50-50% possibility. Even if you take this just one allele and do solve the problem, you will get same 50% blood group as A and 50% blood group as O. Okay. So next question, again same for this, you will get 50% blood group as O and 50% you will get blood group as B. If you want to solve, you can solve as same which I had done here. I will move towards the next question for this fifth question. Okay, what you have to do? You have to do the same. Okay, so what you will get, you can write that I not वगैरह, you can write like this and solve. Okay, I will be little quick now. So you will have AO, you will have BO, you will have AO, and you will have BO. Means what? You will have your A blood group, your B blood group, your A blood group, your B blood group. Means you will have 50% as A blood group and 50% the children will have B blood group out of these two parents. So these are the 5 examples which I have taken, and this is all about the multiple allelism which you should know. Okay, this is all about that. So moving forward towards the pleiotropy. Now what happens in pleiotropy? Okay, we have already discussed pleiotropy in detail in our previous lectures. Just I will give you a glimpse of it, not going, I won't go in detail. So this is pleiotropy. So there is a single gene which is controlling many traits. Okay, many traits means many possibilities. Like suppose I told that there is a trait means what? Suppose there is a height, character is height, and trait is what? Dwarfness, tallness. There are 2 traits. But suppose there is some condition where there are 3 traits. Now we will take example for pleiotropy. Pleiotropy is the phenomena. Okay, we can also say it as pleiotropism. Now there is single gene which is showing many traits. This is the ultimate idea about pleiotropy. Now if we take the example of sickle cell anemia, so there is the gene for sickle cell anemia. This is one allele. This allele is for, like, it is a healthy gene, means you can say a normal, or we can say it as a healthy allele. Okay, and if we see about speak about HBS, then this is allele for sickle cell shape of RBC. Now for which is this, like this is for RBC, and sickle shape is like this, like half moon shape, like not half exactly half, but less than half. I will make it in red. Okay, so it will look like this. So this is for sickle cell, and if there will be heterozygous, so there will be sickle cell carrier. Okay, if you take it as HBA, and if you take it as HBS, then there will be sickle cell anemia. Okay, and if you take HBS, then there will be you can say sickle cell anemia carrier. Okay, and this is the cross which we have to do. Suppose there are 2 organisms, means one female and male, they are mating and they have the heterozygous condition for, means they are the carriers, we can say sickle cell anemia carriers. For this, let us make a cross. So you will have your 2 gametes. Okay, I'm not showing that step. You should understand now. You can draw that like this, showing 2 gametes and doing that all. But now I am doing it directly. So what I will get HBA as the first possibility. Okay, and then I will get the second possibility as HBS. Then I will get the next possibility as HBS. Then I will get the next possibility as HBS. Okay, so here if you can see, this will be a normal, or we can say a healthy organism. Okay, this will be a healthy organism, means the cell RBC will be normal. Okay, this means what? The RBC shape is normal. Shape is normal. But if we come towards this, so there will be a carrier condition, sickle cell carrier condition. Means this is just a carrier in terms of genotype, but phenotype wise it will be normal. Okay, there will be no issue to this person. Then if we consider this, then this will be a sickle cell anemic patient. What will happen? Its RBC size will be like moon shaped. If it is like this, if the RBC size is like this, then what will happen? The amount of O2, okay, means hemoglobin here will be reduced. Obviously hemoglobin will be less, space is less, hemoglobin will be less. If hemoglobin is less, then oxygen supply will be decreasing. If oxygen supply will decrease, then soon the person will die. Okay, then this condition is a lethal condition. What we say this condition as? This condition is a lethal condition. Okay, lethal means dead. Okay, this person will eventually die. So we say this condition as lethal condition. So there are how many conditions? Actually, there are three conditions. One is normal, healthy person. One is carrier person, which is not having any issue. It's just living normally. Okay, but little bit. See, issue will be there because it is having one allele for that, but not that much issue. Okay, the issue will be lesser than this. And the third effect is what? Death, or we can say the lethal condition, means sickle cell anemia patient will die. So there are how many traits? There are total three traits. Okay, there are how many traits? Total three traits. If you see the ratio, we will get ratio as how much? We will get ratio as 1 is to 2. Why 1 is to 2? Because the person who is dying, we don't calculate this. We don't have to calculate this person in the ratio. We just have to calculate that normal is to carrier. So this is the ratio for normal is to carrier. Okay, what is this ratio for? Normal is to carrier. So we have just one normal, we have two carriers. So 1 is to 2 is the ratio. This is a for sure MCQ question in your exam. Okay, I will mark it very important and I will write it here that this is for sure MCQ question. You should know that what will happen if the person is sickle cell anemic. You should know that normal RBC shape is biconcave. Okay, what is the normal RBC shape? It is biconcave. Okay, now the person who are carriers, they get one benefit. See, we know that there is a disease known as malaria, and for this disease there is a plasmodium vivax, or there are many, we can say plasmodium species. There are many species like this is the pathogen, we can say. And this goes and stays in first in liver and then in RBC. Means its life cycle also includes liver and RBC. If there is like a small place, okay, if the RBC size is like little bit sickle cell or something, so what will happen? The size becomes smaller. So the size of RBC is smaller. RBC. Then what will happen? This plasmodium species won't get any space to stay. So the possibility to get, to get malaria, so the possibility to get malaria will also decrease. So they get resistance towards malaria. Resistant towards malaria. And this is actually a very good possibility because in African areas, South Africa and all that, American Africa area, there the chances of malaria is higher. So for that, in the course of evolution, we can say this is the condition which is happening. Okay, they are getting adapted. You can see the adaptation is at the gene level. So this is a helpful thing. So you should know that the benefit which the carrier, okay, which this is what we are speaking about, carrier of sickle cell anemia. Sickle cell anemia. So this is very important that they are getting resistance towards malaria. What they are getting? They are getting resistance towards malaria, and this can be a MCQ question in any entrance exam. Okay. See, carrier is having what? It is having HbA and HbS. So it is being protected. And suppose if the person who stays where at the location where there is more malaria, like disease spreading, so if that person is HbA, so this person will die with what? Malaria. This person will die with malaria. Okay. And if suppose the person is HbS, so this person is will die with sickle cell anemia. Understood? That HbA will die with malaria, and HbS will die with sickle cell anemia. I am speaking about this person when this person is present at the location where malaria is a biggest degree, a biggest disease. So this is this discussion about it. So today's class we spoke about, like we spoke about the so many topics, like we'll go just once. We spoke about codominance as one topic. Okay, then we discussed this cattle coat color and all. Then we spoke about intragenic interactions. Then we spoke about multiple allelism. We did so many examples of blood group and all. We discussed about Drosophila also. Okay, then we came to pleiotropy as a topic. We can also say pleiotropy as pleiotropism. Okay, and the gene which is responsible for this is known as pleiotropic gene. Okay, understood. So this is entirely about pleiotropy which I had discussed. Here you should know that all things which I told, that this is important MCQ. Okay, you should know that where plasmodium is actually going, it's going in liver and it's going in RBC. You should know this. So this is about today's class which I wanted to take in the lecture number 10. In upcoming lecture we will take chromosomes as a topic, and I will elaborate about the chromosomes. We have done with the linkage, so in the next lecture I will just teach chromosomes and Morgan's experiment in linkage. That's all. I will just take two topics in the next lecture, and in the last lecture, that is 12th lecture, we will go with the inheritance and sex determination. Okay, and genetic disorder. These three topics I will take, and in the 13th lecture we are going to solve questions. So that's it for this time. I hope today's lecture is understandable. Do write some comments. Thank you for watching this class.